Hello guys, welcome to Dr. Science. So this is my third part regarding the innate versus adaptive immunity, and in this part I will be mainly talking about adaptive immunity and what are the cells that will come under adaptive immunity. Please watch my part 2 video if you want to know about innate immunity and what are the different types of receptors and cells which will come under innate immunity. Okay. Before going into the topic, I will just give you a brief overview of what is going on. Okay. So here is adaptive immunity. Adaptive immunity is divided into two types. One is humoral immunity and the second one is cell mediated immunity. So in this humoral immunity is nothing but the immunity where antibodies are produced. Okay, you see antibodies in this type of immunity. So how are antibodies produced? We'll take a look at it. Suppose, let me draw a clear diagram. Suppose these are the B lymphocytes, and B lymphocytes in the presence of T helper cells, they converted into plasma cells. Okay, and these plasma cells will produce different types of antibodies like IgG, IgA, IgM, IgE and IgD. Okay, so this is what humoral immunity means. So what are the cells which will come under humoral immunity? B lymphocytes, plasma cells and antibodies. Okay, the next is cell mediated immunity. In cell mediated immunity, we will mainly see T cells. Okay, T cells are mainly divided into two types, or we can say three types if you want to know. The first one is T helper cells, the second one is T regulatory cells, and the third one is T cytotoxic cells. Why these are called as T cytotoxic? They will kill the cells. Cytotoxic means they are very toxic for the cells. That's why they are called as T cytotoxic cells, and these kill the cells which are damaged or infected with viruses. Okay, so T helper cell, they help in the conversion of B lymphocytes into plasma cells. Okay, all these comes under cell mediated immunity. Okay, so this was about humoral and cell mediated immunity. Next I will talk about how these lymphocytes mature. Okay, so suppose this is the bone marrow. In this bone marrow, this is the bone marrow, and in this bone marrow there are cells called as lymphoid, since lymphocytes come from lymphoid progenitor cells, and these lymphoid progenitor cells are immature. Okay, and they have two options: either they can be converted into progenitor B lymphocytes, or they can be converted into progenitor T lymphocytes. Okay. B lymphocytes, they mature in bone marrow. That's why they are called as B lymphocytes. B stands for bone marrow. In bone marrow they get mature, whereas T lymphocytes, progenitor T lymphocytes, they get matured in T lymphocytes. T stands for thymus. Thymus is present behind the sternum, so that's the place where they get matured. So that was about the maturation of B and T lymphocyte. Next we will see of what is happening to the T lymphocyte. So suppose this is a lymphocyte, and these lymphocytes have certain receptors present on it, and suppose there came a virus called coronavirus. So here is coronavirus with red receptors. So what will happen to these lymphocytes? They cannot attack the red antigen. This is the red antigen, hypothetically speaking. This is the red antigen present on the coronavirus. So what these lymphocytes do? These lymphocytes, they have this proliferation capacity. So what they will do? They have a different type of proliferation. So suppose these lymphocytes are having red receptors, and these lymphocytes here are having green receptors, and here are another lymphocytes having suppose blue receptors. So we want red receptors because the coronavirus is having red type of receptors, right? Red type of antigen. So we want this. So what the body will do? The body will activate this clone. This is the clone here. So this is the clone, and this is called as clonal selection. This is clonal selection. So among these three clones here present here, we want a lymphocyte which has red type of receptors and which can attack the coronavirus. So this is called as clonal selection, and once this clone is selected, they get proliferated. Okay, they again get proliferation, and here are the daughter cells which will have more type of red type of receptors. Okay, and these are called as clones of each other. Okay, so that was about clonal selection, and these are the clones of each other, and the cycle goes on. There will be like lot of proliferations going on, and there are many daughter cells against which the coronavirus will fight. Okay, so next we will talk about immature cells, immature lymphocytes. Suppose this is a lymphocyte here, and this lymphocyte is immature because there is no antigen presented to it. This is the antigen. Okay, antigen is not presented to it, and it is immunologically inexperienced. It means it is like a teenager where there is no experience at all. These lymphocytes are called as naive lymphocytes. Okay, which are immunologically inexperienced, naive lymphocytes. So what happens to these lymphocytes once their antigen comes and binds to this receptor? Now they become like a little experienced. Once the antigen is presented to these lymphocytes, they become like experienced, and now these cells are called as effector cells. Okay, not only that, these will form daughter cells. Okay, these daughter cells here is a daughter cell, and here is a daughter cell. These will remember the memory. It means this will remember, okay, this coronavirus has spike proteins, and they have these are called as memory cells, and compared to other cells, they have heightened immunologic experience, and they are like 24 hours active. They are waiting for the virus to come, and they are ready to attack that type of virus. Okay, these are called as memory cells. So this was about adaptive immunity. Now we will take a look, we'll go deeper into that. So here are some of the cells I told you. So this is a B lymphocyte. B lymphocyte comes under humoral immunity. So B lymphocytes microbe is presented, and this is the plasma cells here, and these plasma cells are producing antibodies. Okay, and these antibodies will go and bind. These are the microbes here, and these microbes are presented with antibodies which help in the neutralization of microbe phagocytosis and complement activation. I will explain these in the next videos. Okay, next we have T helper cells. So B lymphocytes in the presence of T helper cells here get converted into plasma cells, as you can see. Not only that, T helper cells are T helper cells there are different types: T helper 1, T helper 2, and T helper 17. The function of these T helper cells is activation of macrophages. Macrophages are activated. Okay, I will explain this one in the next video so you don't get confused. And the second one is recruitment of neutrophils. So here are T lymphocytes, and they are activation of macrophages. Obvious, once the macrophages are activated, they will cause inflammation, and they also do the T and proliferation of T and B lymphocyte. Next, cytotoxic T lymphocyte. They come under cell mediated immunity, right? And what do the cytotoxic T lymphocytes do? Yes, cytotoxic stands for killing of the infected cell. So here is killing of infected cell. And what does the regulatory T lymphocytes do? They help in suppression of immune response. This one is very important, because whenever there is a virus or a pathogen is attacking, the T cytotoxic cells are very toxic to the cells, and we prevent this by suppressing the immune response by T regulatory cells. And natural killer cells are, no, they don't come under adaptive immunity, they come under innate immunity. But in this picture I just included it. So what is the natural killer cells do? Natural killer cells and T cytotoxic cells, they do the same thing. They do the killing of the infected cell. Thank you.